Okay, what was the question?
First one.
The first one, what was the first one?
Interface accounts for 90%.
90%. Yeah.
90%. Interface accounts for 90%. So how often is a cell in the G0 stage?
10%.
10%.
Really? Because the cell cycle at any given time is only 10%. So 90% of 10% is
9.
So that means 9% total is in interphase, 1% is in end phase, and so that means most of the time, 90% of the time, the cell's going to be in this G0 state, in the undivided state. So most of the time your cell's just sitting there, not doing anything. Well, I mean, it's doing stuff, but it's not replicating its DNA and making copies of itself. And so the other 10% of the time is when it's in the cell cycle. And of that 10%, 90% of that, or 9% total, is in interphase. And 1% is in end phase, total. So at any given time, a cell is in end phase 1% of the time, or it's in interphase 9% of the time. Other questions?
What is the purpose of the G1 checkpoint? I'm gonna go down.
Oh, we are not going to go down the list. I got better things to do with my time than sit here and answer all the review questions for you. That's why I took the answers off, because last time I gave you the answers and you still didn't do that hot on the test.
I did better.
So you're going to learn better. You're going to learn better if you look up the answers yourself. So I'm not. Yeah, I'm not going to sit here and answer all the questions for you. That's not my job. I've already taught you this stuff. It's your job to learn it. Question? Anybody?
Yes. What is the central dogma of biology? Central dogma?
Yeah, so that's basically just talking about the order of events in gene expression. So DNA gets transcribed into RNA, and then RNA gets translated into protein.
That's central.
That's
So
simple.
that's the simple answer?
Yep, that's the simple answer. DNA to RNA to protein.
Why is it called dogma?
I mean,
That's what
if-
that threw me off too. It's like, just,
Yeah, it's just called the central dogma. I mean, it's just like dogma is something that's like been ingrained for so long, so it's a dogma. It's kind of like a law or like a theory or I don't know. It's just the central dogma. Another way to phrase the question would be, what is the order of events in order to make a protein? Same question, same answer. Question? I heard something.
I still got the same question.
What was the question?
What is the purpose of the G1 checkpoint?
Well, okay. Well, let's look at the cell cycle. We got G1, we got S, we got G2, and we got end phase. Where do you think the G1 checkpoint's going to be?
before.
After the G1?
After. So what do you think the G1 checkpoint's purpose is? To make sure everything's okay before it moves forward into S phase. So particular, because the cell is going to enter right here in G0, there's no point in having a checkpoint at the very beginning if he's already going into the cell cycle. And so once he goes through G1, then there will be a checkpoint to make sure that the DNA is all nice and pretty and ready to be replicated in S phase. If it's not, then it'll kick it out until it gets fixed, and then it can come back in. Yeah, Michelle?
So, at each checkpoint, like the G1 checkpoint kicks it back out to G0. If it gets to the next checkpoint, does it kick it all the way back out to G0?
Yeah, if something's wrong at any of these checkpoints, it'll automatically kick it out to
So
G0.
back to the
And then once it fixes itself, wherever it was left off, it'll come back to that checkpoint.
Okay, so it doesn't go back.
It doesn't have to go all the way back through. Yeah, it can just come back in where it was. You know, it's like waiting in line at an airport. You get pulled over for security, you get checked, you get to move forward. You don't have to move back to the end of the line again. All right, Candace?
So G1 is literally just make sure it's like, it's all right, like S checkpoint would be to make sure it replicated correctly?
Yeah, exactly. You're making
So
sure that the
G1's
step
just-
before it went according to plan.
It's just in general, it's okay?
Yeah, it's just a generalism. Yeah, so the G2 checkpoint comes after G2. and make sure everything here went okay. The M checkpoint comes after M and make sure everything in M was okay.
So that G checkpoint is DNA damage. You know, if not, it'll go out and you'll come back in.
For this one, because it's preparing, don't worry too much about, just the basic point is that it's there making sure that whatever previous phase went okay. And everything's ready for the next phase. So for example, at the G1 checkpoint, you're getting ready to replicate your DNA, right? So this is making sure that your DNA is ready to replicate. Just like here, you're getting ready to divide your cells, so you gotta make sure your cell's ready to divide.
And what about half as much DNA as some other cells?
So read the question for me.
A particular cell has half as much DNA as some other cells in I can't pronounce.
All right, so a cell has half as much DNA as another cell, and they're both in the cell cycle, right?
Yes.
Can you read it to me again?
Mitotically active tissue.
In mitotically
Mitotically.
active tissue. What do you think that
The
means?
cell in question is most likely in.
Okay. So we're talking about mitotically active tissue.
So is that in mitosis phase?
Exactly. That just means it's undergoing cell division at some point. It's mitotically active. It means it's dividing.
But we need to locate exactly where in mitosis, so.
Right. So you use the context clues. So the question is, you have a cell that has half as much DNA compared to another cell that has the entire amount of DNA.
Is that anaphase?
So we're talking about the cell cycle. It says what phase of the cell cycle is it in. So there's only 4 options. G1, G2, S, and M.
Well, it says prophase, metaphase, anaphase. are options also.
Well,
If she's not gonna be that specific, y'all don't be that specific. Just
G2.
thank you.
M is great, that stage.
Yeah. Okay, so if you have a cell that has half as much DNA, so you have 2 cells, they're both in the cell cycle, which means they're both mitotically active. But one cell has twice as much DNA as the other. So which stage do you think the cell that has half as much DNA is in?
Anaphase?
Not anaphase.
Prophase, G2?
Not G2.
I'm sorry.
All right, where do we double our amount of DNA?
S.
S phase, right? So the cell that has half as much DNA has not gone through S phase yet. So it's in G1. Whereas the cell that has, the full amount of DNA, it's somewhere after S phase. So the key is half as much DNA. And if you know that in S phase you double your DNA, then you should be able to deduce that before that happens, you have half as much DNA. That question is on the test.
Yes.
That exact same question.
Is a short answer.
No, it's multiple choice.
What happens during binary fission?
So binary fission is the way that bacteria divide. It's actually a precursor to mitosis. It's just real simple. All that happens, you've got your bacteria. Remember, bacteria don't have a nucleus, so they've just got all this DNA in here. So the first thing that's going to happen is it's going to replicate its DNA. So just like in normal human cells or eukaryotic cells. So it'll replicate the DNA, and one copy will attach to this edge of the cell wall, and this copy will attach to the opposite edge, and then this cell starts to elongate, and as it elongates, it pulls that replicated copy of DNA with it. And then the last thing that's going to happen is, similar to something like telophase or cytokinesis, you're eventually going to get those 2 cells are basically going to pinch in half to make 2 cells.
What about the Chargaff's rules?
Chargaff's rules. So this is the very first thing that we talked about.
Okay, maybe that's wrong.
Yeah, the number of A's is going to be equal to the number of T's, just like the number of C's. It's going to be equal to the number of G's. And the reason being, A's always bind to T's, and G's always bind to C's. So you could also throw in there your cells as well. So if I gave you a question that said a cell has 70% G's and C's, What would be the percentage of Gs?
30%. Or 15%.
Like 35%.
What's half of 70?
35.
35, right? So that means 35% has got to be G and
Oh, gee, I
35%
thought you said T.
has got to be C.
I thought you said T.
Maybe I did, I didn't mean to. But, so, based on this, how many, what's the percentage of A's?
15.
15, right? Because the difference is 30, so that's got to be split evenly between A's and T's. Yeah, Nancy?
On the mutations, it
Uh-huh.
says describe silent, the missense, and nonsense.
Uh-huh.
Is silent the duplication?
No, the silent is where you're still changing the DNA, and it's still going to affect the RNA, but you're putting in the exact same amino acid that was supposed to be there. And so remember when we looked at that codon chart, that there's multiple different codons that can code for the same amino acid. And I'm just kind of making this up, but so I don't know. No, that's a stop codon. I can't use that. All right, let's just say C, 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 G, C, C, A, and C, C, T, all code for the same amino acid, right? Whatever it is, I don't know what it is. But, so if you mutate this, basically what that means, it's a mutation in this third base pair is what that means. So it doesn't matter if there's a C here, or an A there, or a T there, as long as there is a C in the first spot and a C in the second spot, you're going to get that amino acid. So it's still a mutation in the DNA, and you're still changing the codon. It's just, you end up coding for the same amino acid that you would have in the first place. And that's because of the redundancy of the genetic code, meaning there are multiple codons that code for the same amino acid.
So with silent, even though it has the mutation, it'll still come out with the same.
Yeah, no harm, no foul. Still technically mutation because you changed the nucleotide, but it just so happened that you changed the nucleotide in the third codon set, which, if all of these code for the same amino acid, it doesn't really matter.
Okay. I was just trying to compare that to the illustration that you gave with the types of mutations with the.
Oh, with the
Sentence.
big, okay, so let's,
So,
I'll
would
just
it be
make
on-
up, I'm not good at making up random stuff like the book is, like the one big eye, whatever. So I'm just going to use a real example. So let's just say I have a piece of DNA. Hold on, let's just say my DNA is, I don't know, A, T, G, A, C, C, C. All right, there's my DNA. So my RNA is going to be U, A, G, U, 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 G, G, G. So the amino acid for this is the start codon, this is phenylalanine, and this is, I really have no idea what it is, it's some amino acid. So if I get a mutation, let's just say I mutate this right here, and I change it into a T. Well, then that's going to change this to what? An A, right? And so the difference between amino acids is if I'm looking at the codon for this, if all the codons that start with GG all make the same amino acid, then it doesn't matter what that last codon is. So by changing this CCC to a CCT, I'm still coding for the same amino acid. So it's a mutation because this was changed, but it's not affecting the protein because you're putting in the same amino acid. Now, a missense, that's where you do change the amino acid sequence. And then a nonsense, that's where you generate an early stop codon. So a mutation causes you to make either a UAA, a UGA, or a UAG. Yeah, Daniel?
I don't know if this is on the study guide or whatever, but you know you have your DNA strand and your DNA replication.
Uh-huh.
Which one is RNA copied from?
Which one is RNA copied? Oh, when you're making RNA? So it depends on how you do it. So let's just do another example. Actually, let's just use this example right here. Okay, so here's my DNA, ATG, AAA, CCT. And so one strand will be the 5' to the 3', and then the other strand will be the 3' to the 5'. This strand right here, this 5' to 3', this is called the coding strand. And then the 3' to 5', that's going to be the template strand. And so our complementary DNA here would be T, A, C, T, 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 G, G, A. So if you're taking it from the template strand, in other words, the 3' to 5', all you need to do to make your RNA.
Are you always going to take it from the template strand to make RNA?
The way that it's going to be on the test is I'm just going to give you one strand. I'm going to make it simple for you.
Well, I'm asking because of my lab too.
Okay. Well, so if you remember the homework question, it was like use the coding strand. So if you use the coding strand, then you're putting in the opposite. So if we were using this, our mRNA would be U, A, C, U, 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 G, G, A. So that's if you're using the coding. If you're using the template, then that's where you just put the opposite. So this would still be 5' to 3'. You put the opposite, or sorry, you put the exact same.
As the DNA, just put in the U's.
Change the T's to U's.
Yeah, change the T's.
I thought you copied the coding and
You copy, I'm getting them mixed up.
you copied the coding and you
You do the opposite of the template.
yeah.
Teachers.
So
Okay.
when you use the coding, you end up with
It doesn't matter. It doesn't matter because I'm only going to give you. I'm only going to give you one code. But if you use the 3' to 5', all you do is you put the exact same thing. So if you're using this one, this is going to be your RNA. If you use the other one, then that's when you're doing the. You can't do the opposite because it's the same thing.
Right.
yeah. You copy the coding
Totally true.
to coding and you translate or you swap templates.
Exactly like we did.
If you did the coding for mRNA, it would be AUG, AAA, CCT, or CTU.
Yeah, because your mRNA automatically has to start with AUG.
Right, yours doesn't.
Yes, exactly. So that's why.
Yeah, you
Sorry
got
to be blunt, but it doesn't.
No, I know it doesn't. That's why. So if it starts off with AUG, that means the template has to be, or not you, TAC. And so if you're going to put in the opposite, this is the template.
Which one are you going to give us on the test and which way do we need to
I'm
do
going to
it?
give you one piece of DNA. I'm not going to give you double-stranded.
But will it be
For example.
Yeah, but would it be the
For
coding?
example,
Sorry. My bad.
make a piece of RNA.
Okay, so you just take the T's out?
Just put in the opposite. That's all I want you to do. I want you to say
Dr. Gill,
A, U.
I asked something totally different.
Oh, you did?
Yes.
Oh, well.
I was talking about,
All right, my
because
bad.
when we were in lab, we did the assignment or whatever in the book, and it said like, there's a DNA strand column, and then next to it, it said DNA replication, and it had, the nucleotides going down. So next to that column, you had a column where you would, make the complementary strand of it. Thank you.
Okay, so DNA replication.
And you see on the far left column.
Yeah, so it gives you the strand of DNA and it wants you to replicate it. So you just put in the complementary
Well, I
nucleotide.
didn't know which side we were complimentary.
Oh, I see what you're saying. Okay, so it still kind of goes to this. This doesn't go into whether it's a template or, well, normally if what it gives you, this would be like what I gave you. So this would be the DNA strand. So if I said to replicate the DNA, it would be the opposite. It would be AAA, T, T, T.
Now make RNA.
Now make RNA.
Replicated, right?
It has to start,
TTTU.
yeah, so all DNA has to start with this TAC. So this TAC, this is going to be the 5', And this is going to be, actually, this is gonna be the 3' to 5' because you can only add nucleotides to the 3' end during replication or DNA synthesis, or during transcription. And so you're always gonna use the 3' to 5' strand to translate, because when you do, or when you, whatever the 3' to 5' strand is, you just copy it. So it's UAG. If I gave you the 5' to 3', that's when you put the opposite. So you're still getting this AUG.
It's supposed to be AUG.
So the
Yeah, AUG. AUG.
The RNA should be starting with
RNA will always start with AUG.
3'.
So that means your 5' to 3' strand of DNA will always start with ATG. And so if you're using the 5' strand, you just copy it and just replace the T's with the
You're just
U's.
gonna give us a DNA.
I'm going to give you this piece of DNA. I'm going to give you the 5' to 3'.
And then
And we
we
have to
flip it
just-
and do 3' to 5' on the RNA.
It'll be very simple. Y'all are overcomplicating this. I'm not even going to put 5' and 3'.
Write some DNA on that.
Okay.
And show us what you want us to do on the test. I'm completely confused on what we're doing on the test.
Okay. I'll say here's the template strand.
So it's the 3'.
Make a copy of DNA. So there's a template. So my new piece of DNA, GCCAAT. Now say make a piece of RNA from the template strand. So if I say make a piece of RNA from the template strand, then GCCAAT.
You.
I
You.
mean, yeah, sorry, you.
And you wonder why we're confused.
No.
I think you should just pass it.
Yes, do that.
Because that wouldn't be
$10.
illegal or a
From
bribe.
each of us, or you did, dang.
Not that much money.
I don't know, there's like 20.
I don't know, I can pay more per hour.
I'll wash your car for you.
I do need a car.
I mean, you got a car wash.